Good evening to all. Today, the resource person is Dr. S. Padma ma'am, Head Department of Biochemistry and Nutrition, and also the course coordinator for this value-added course, Bioinformatics Tools in Genomics and Proteomics Study. Ma'am has a rich teaching experience of 24 years. Ma'am has dealt with wide subjects like bioinformatics, clinical biochemistry, endocrinology, biostatistics, and many more. Ma'am has 16 number of publications in reputed journals with 169 citations to her credit. Ma'am was also a recipient of Postdoctoral Research Award in 2012, where they have granted or sanctioned an amount of rupees 14 lakhs for the project, which was being funded by UGC, University Grants Commission. Ma'am also has been a resource person taking bioinformatic classes for the postgraduate students of Institute of Genetics, Hyderabad. And also ma'am was a resource person for teaching the research scholars at Osmania University. Ma'am's rich knowledge and expertise in the field of bioinformatics has encouraged all of us and supported us in framing this value-added course content. Today, the session is on phylogenetic analysis of protein and nucleotide sequences. Padma ma'am, welcome to you, and we are eager to learn from you. The platform is open to you now, ma'am.
Thank you, Anita. Good evening to all the participants. I hope you have revised and come afresh with your knowledge of cluster omega sequence alignment. Yes, students, participants, can I have some response, thumbs up or say yes?
Yes, ma'am.
Yeah.
Yes,
So,
ma'am.
so yesterday's class, you had a brief, you were explained about the sequence alignment, what is multiple sequence alignment, and what is the importance of that multiple sequence alignment, how to identify the conserved residues, and several other aspects were explained to you by Dr. Vanita. So with that background information and what further we can, and also you were informed yesterday, Vanita ma'am had explained about a guide tree and a phylogenetic tree. So you have to refresh your memory what we have discussed in the previous class so that we can go further today. So kindly mute yourself, participants. Please, Nikita, please mute yourself. Nikita, Nikita, please mute yourself. Nikita. Okay, so let me first share the screen. Is the screen sharing?
No,
No,
ma'am.
ma'am. No, ma'am.
Yes, let me try once again. Is it sharing now?
Yes,
Yes,
ma'am.
ma'am, it is.
Yeah, thank you. Okay. So today I will be telling about the phylogenetic analysis of proteins and nucleotide sequences. We'll start with nucleotide sequences and we'll see whether we can complete about the proteins also. So, phylogenetic analysis. What is meant by this term, phylogeny? Anyone? Ontogeny, phylogeny, genetic students, or anyone in the group?
Study of evolutionary history.
Yes, ontogeny repeats phylogeny. You are aware of that statement?
Yes, ma'am.
Yeah, so we are going to discuss about this phylogenetic analysis. Uh, so when we go to this term phylogenetic analysis, let us see what it tells us, why it is important for us as far as our genes or proteins or nucleotides are concerned. So, what does it tell us? Analysis means if you are taking a sample, protein for instance, or a gene for instance, there are several genes which have been conserved throughout evolution, and uh, when these genes are conserved throughout evolution, we would be interested to see what could be the relationship between various species. If you take a simple gene which is there from the lowest microorganism to the highest species like Homo sapiens or any other mammalian species, some genes are conserved over the period of time, and we would like to see whether these genes are conserved across which species, and which species is related to which other species. Why it is going to be important is uh, to know that evolutionarily which are the organisms that are closely related to each other, and which organisms are distant from each other. Okay, so if you are looking at this particular example here, you can see this is a conventional tree, whether whoever it may be, without life science, without bioinformatics knowledge also. Let us just discuss, or let us just consider this as a plant or as a tree. So a tree is something which would be rooted down into the ground, and it would be having a thick stem, a bark, and then the bark leads to the stem, and the stem leads to the branches, and then we have the final leaves coming on the branches, and the tip of the branch tends in the leaves. So here, in the same way, from where are the origins of that particular gene? From where is it originating? What is the organism from which it is originating? To understand that, we take this particular analysis, which is very important as far as the genes are concerned. If you take this example, you can see that it is a common ancestor which is there, and this common ancestor is giving rise to diverse species, which may be fungi, which may be plants, and there is a central branch which is giving rise to most of these vertebrates, invertebrates, as well as the mammals. So you have insects which belong to arthropoda, fish belongs to Pisces, reptilia, amphibia, so, and then mammalia and aves. All these, if you see, these are all in a different evolutionary branch. Plants are also in an different evolutionary branch, and the fungi are in a different evolutionary branch. So what is the basic difference between all these three types of living species, fungi, uh, vertebrates, invertebrates, as well as the plants. So these uh, type of relationship, if you see any example or any gene which is distributed in all these species, you would like to know what sequences are conserved, and what possibility is there that a plant species can be evolved from animal species, or an animal species could be evolved from a fungal species. So that's why this becomes uh, analysis which represents the relationship amongst all the sequences. And this is also, for example, used to follow the changes occurring in a rapidly changing species such as a virus, which is the most common example all of us talk about. Even after so much, after a long time, though it COVID has come and gone, we still work with those genomes, we work with those virus, and we know that this particular species virus is continuously changing its genome. There are wide variants, and recently also there has been a scare about similar viral outbreak, and different species are exposed, or different species are being released into the environment, which may be harmful to the population. So in such a case, what is the way you can analyze the genome and find out what is the mutant varieties which are coming up, and why you can call it as a mutant, and if it is a mutant, what could be the difference in the sequence where the nucleotides are differing, how crucial those nucleotides are, and what does it translate to? That means, since we know that DNA sequence is important, and DNA later gets translated into the protein, and whenever the letters nucleotide sequences are changed, it manifests as a mutation of the amino acid. So these thing, all these are interrelated to each other. Whether DNA sequence is important or amino acid sequence is important. Is it clear? So if I ask you the same question, if I ask the participants who are here, which do you think is important? A DNA sequence is important, or a protein sequence is important?
DNA.
Why so? What makes you think? Can you justify your answer why DNA is important?
Because the process of this DNA, first DNA, then it will come mRNA, and then protein. DNA is the main.
Yeah, DNA is main, but also you have to remember that our genetic code, which is the nucleotide sequence, which is going to the protein, is also having a degeneracy. That there are codons which are coding for each amino acid has more than one codon. So in that case, what is your answer? If you say the particular amino acid, is it that amino acid has only one codon in our genetic code? It is a universal genetic code at any species, whether it is a fungus or it is a plant or it is an animal, it is a universal genetic code, is it not?
Yes, ma'am.
So, so there may be some, if the change occurs in a triplet codon, if the change occurs in the third nucleotide position, then it may not be leading to a deleterious mutation, or it may be not, it may be not causing a harmful mutation. But if someone please switch off your, please keep yourself on mute. Yeah. So, so DNA is more important, as you all said, DNA is very important because though if any change occurs in the first nucleotide or the second nucleotide position in a triplet codon, then it is a deleterious change. So now here, what we can understand is what is this phylogenetic tree construction, how it is going to be giving us information about the evolutionary relationship. In this, you can see that this is a sequence which is from the human sequence. Now this human sequence is being compared to animal and also compared to a fish genome sequence. What you can observe here, as you can see here, this human species, human is a mammal, and this is also a mammal. And what are the changes you are observing with respect to the fish, which is relatively in the lower rank in the evolutionary uh, process? So here in this, you accept these three nucleotides CTC, which is here, and this particular G, which is conserved. There is no other similarity in this sequence compared to this sequence. If you compare the human sequence with an animal sequence, you can see that there is a out of these nucleotides which are here, there is change with respect to only two positions compared to the fish, which is having more number of changes. So what is it that we are concluding from this kind of analysis? We conclude that these two species, they have close resemblance, they have a common ancestor which is shared with this fish, which fish has come out of this group because there are some group changes which are there that have taken place in the sequence. And because and so this particular set of sequences which have very limited variation in their nucleotide position, they can be grouped together, while this particular species which is having variation of several nucleotides, that can be categorized as a separate group. So this tells us evolutionarily this particular species is not directly related to these two species. However, these two have close relationship because they are having by visually inspecting this, you are looking at these two sequences, visually inspecting and seeing that okay, there are the similar residues here. So because these similar residues are there, these two species are together. Then why this one, why are these branches here, what does this signify? And you can see how much what in what basis you would give this kind of relationship between the species. Okay, clear, students?
Yes, ma'am.
If any question or any query is there, please ask me. Okay, so let us briefly see what these phylogenetic trees are. So to define that, molecular phylogeny is a study of evolutionary relationship among organisms, that is different organisms are there, or among different types of genes which are there using the techniques of molecular biology. And why do we study this tree construction? In order to understand the evolutionary significance which can be related to their physical appearance, that is morphology or anatomy. The anatomy is with reference to the body parts. Can you give me some example related? When I say anatomy, can you relate some anatomical parts with the example what I have shown you? Between the in this example what I have shown you, can you tell me what is anatomical parts? Yes, anyone? What is common between three
Legs.
what is the common thing which is there between a Homo sapien, an animal, and a fish?
Eyes.
Yeah, eyes.
Mouth.
Okay. Then Limbs are there in humans and animals, and which are used for movement, whereas the fish has what? Tail and the fins, which would help it to propel forward. So like this, there are many uh, anatomical similarities, and physiologically, can you tell me what can be the similarity physiologically?
Breathing.
Yeah, respiration, digestion, absorption, excretion, all these are the various physiological activities which are there. So, what are the common features, or evolutionarily how these events are related? So, to understand that, or to make that, we take the help of these phylogenetic trees. So, these trees, they use molecular sequence data. These phylogenetic trees also provide information regarding the topology, that is, if there are two homologous protein sequences, what is the common ancestor of these two homologous protein sequences? And to understand this, we have a branch length, which is nothing but the degree of relatedness of the objects which are there in the tree. So let us see the terminology here. What is the terminology which comes across when we go into phylogenetic tree construction? So the important terminology which you can see here is a branch. As you can see here, the branch is one terminology. Node. This node is of two types. One type is the internal node, another is the bifurcating node, and an external node. So you have three types of nodes. If you observe this phylogenetic tree, you have terminologies like what is a branch, what is a node, these two bifurcating, what is the difference between a bifurcating node, internal node, and an external node, right? So bifurcating node is this particular one which is dividing into two, having two other species which are evolving from this particular point. When you consider this node, you relate it with a plant. Plant is having the node from where you get either a leaf or you get a flower. So clad is nothing but a leaf from which arises from the node. So this particular segment where you are having two different species which are sharing a common ancestor, this particular section, you call it as a clad. Clad is nothing but another name for the leaf, right? We know that a node always leads to the uh, leaf, or it comes to a branch or some other part. So in our case, the node is giving rise to this species, this two diversified species, or which is considered as a clade. Now, this branch is divided into two branches. You can see that there is a difference in the length of these branches. What the length of these branches indicate? Remember this particular image where I am showing the length of these branches are not uniform. Length of these branches are not uniform. Why it is not uniform? We will see. So you have to remember every depiction in a phylogenetic tree is giving you some kind of information about the relationship between the species, right? So one we have seen is a bifurcating node. The another one is the internal node. Internal node means this node is going to give rise to some other nodes. All these are internal nodes. And this particular one which ends up terminal node or external node, you can see what is the difference between this and these two species is this is completely away, like the fish what we have seen in our example I have shown you in the slide. The fish is completely away. You can see that In this fish is completely away. So this particular node is called as an external node. So what are the types of nodes we have? We have a internal node, bifurcating node, and an external node. You can see that in this case, this is an external node. If you take this particular window here, in this window, this is an external node. Why this is an external node? This is xenopus. What is xenopus? What is xenopus? What is it scientific name for? Yeah. So this one, frog, this one is different, and you can say gallus or rabbit or horse, pig, all these are mammals, while as this is an amphibian. So this has been completely placed out. So this is an external node. But this one is not an external uh, node here. Based on looking at why this is an external node in this case, because this is pig and these are horse, buffalo, rabbit, and cat or dog. This is a bird. So this one becomes an external node compared to the rest of the members in this group. But there are some, why all of them are categorized in one group? It is because they all share some sequence similarity. The extent of similarity, the extent of genes which are there in them may vary depending on the percentage of similarity. That's why their relationships are different. Okay, so that is about the terminology. So in terminology, we have a clade, node, and a branch. So this section we call it as a phylogram. So how to arrive at this particular image, or how to come to this tree construction? Okay, so let us see what are the types of tree construction that is possible. In what ways the tree can be constructed? Okay, so one is a rooted tree, and the other type of tree is called as an unrooted tree. If you observe the two types of trees here, this particular tree is an rooted tree and this is an unrooted tree. So what is the observation you are making from these two types of uh, trees? So this has a node, this is a node, this is internal node, and this is a completely out node or external node. So there are, this is a parent root, parent sample from which there are two species which are diversifying like this. Now from this particular section, you can see that these C and D, these are having a common bifurcating node. This is a bifurcating node where C and D have come out. Similarly, this is also a bifurcating node which is having A as well as B. Now this A and B are grouped as one clade, C and D are grouped as one clade, and this is connected with the common ancestor. So there may be some amount of similarity when you are comparing the sequence between A, B, C and C, D. Why do you get this type of connection? How is it possible to get this type of connection? Here A and B have similarity, here C and D have similarity. Beyond that, AB may be having some similarity with C, or BC may have some similarity with D. That's why there are some common branch points. For this would be a bifurcating node. This is also a bifurcating node. This is the external node or a terminal node. So this is a rooted tree. It is having a strong uh, ancestral origin, and this is one way of reading the tree. Now, if you see the unrooted tree, this unrooted tree is not, if you see the same samples A, B, C, D, E, here you have seen that A, E is completely out here, so it is an external node. But now, in the case of an unrooted tree, what is it that you are observing? In the case of unrooted tree, these node points are in some way related to each other. They don't tell us anything about the evolutionary significance. If you see this, we can say that this species is completely out of this group and is not having very minimal change or minimal relationship between the other members of the group. But if you see this particular display of the tree construction, which we call it as unrooted tree, there is no information regarding the direction of evolution. It is only telling you that this is one node, this is also one node, what is the relationship between these two nodes and what is relationship between these two nodes. Whether these C and D have evolved or whether A, B and C, D share a common ancestor like this, nothing, no information is given from this kind of arrangement or this kind of depiction. So this is an example of an unrooted tree. This is an example of a rooted tree. Is it clear?
Yes, ma'am.
Yes,
Everybody,
ma'am.
all of you are following? Yeah,
Yes, ma'am.
yeah. So now let us see here. Let us see this example here. Now I have shown you an example with respect to human genome and human, animal and fish. Now that was related to the different types of species, whether it is an animal species, whether it is a fish and an animal. Now here what I as I've told you earlier also, you can use any type of molecules to establish the tree construction. Now this is an example of a rooted tree which is showing different types of globin. Okay, different types of globin. Is it clear? What we are seeing here, there are 9 types of globin here. This is myoglobin, neuroglobin. This is from plant source. This is from birds. This is from mammalian source, human, chimpanzee, dog, and mouse. Right? So this one is showing you with respect to the at the level of the gene. Previously, we have seen with respect to those a particular distribution of the species which are related or which are analyzed. Now here a single example, single gene we are taking, that is an example of a globin gene which is distributed evolutionarily in all the life forms, and it is distributed with respect to tissues, with respect to plants, with respect to plant birds, with respect to mammals. Now how these different, whether you should work with a globin gene which is there from the plants. If you use a globin gene from plants, can you use that gene to extrapolate or relate your findings in the case of an bird or in the case of any other mammalian species? So you have a globin gene in hand and you want to know, if you work with that particular globin gene, whatever you are getting the result, is it holding true? Is it holding true with respect to all other life forms? It may be a plant or it may be a bird or it may be a other species. So what we have here is a different set of globins which have been constructed, used as to make a tree construction. Now you can see here there are some numbers which are depicted on this particular branches. Now as we have seen earlier, this all these are put into one clade. So what is a clade? A clade is a set of sequences that form a group within a tree. Why are these forming a group? You can see the branch length which is here. This is around, whatever these numbers are, we will come back to that. This is 0.06 and this is 0.06, which means that there is a similar type of sequence which is there between the mammal dog as well as humans and the chimp. Human and chimp, they share more sequence similarity compared to the this clade is separated with this dog, and this in turn is different from mouse. Mouse has been completely kept as an external node. Dog is an external node here. So, but these together, they are all mammalian globins. We are comparing this, for instance, with a bird hemoglobin or bird globin gene. You can see this is a distance length which is this value is quite high in distance compared to this particular distance. So this hemoglobin chicken from chicken is an external node, and it is at a distant evolutionary history from this particular set of species. Likewise, if you take a plant gene, you have a globin soybean and a globin rice sequences. Now, if you see this particular example, these two are 0.24, 0.24, 0.25, which are coming from a common ancestor. This is the common ancestor which is there. This has gone into a, a chicken as well as giving to the animal cells, and another evolution has happened which is giving to the globin in the case of plant species. Now this root is here. The root is the one which represents the most recent common ancestor of all the sequences. So here again you can see that this particular myoglobin and neuroglobin, these myoglobin is a completely present as an external node compared to the neuroglobin. These two are not they don't have much sequence similarity because they are not in the bifurcating node. They are separately present here. Or so like that, when you look at this particular data, this is a rooted tree because it is having a common ancestor which is giving rise to the other sequences. Is it clear?
Yes, ma'am.
Yeah. Now, the coming to the unrooted tree. An unrooted tree, I given you the example of an unrooted tree. An unrooted tree is the one where the relationship between the nodes is not clear. It does not give us any evolutionary relationship. Now the same set of sequences which I have shown you in the previous slide by rooted tree, here it is shown by drawing an unrooted tree. So it does not define the evolutionary path. In a rooted tree, we have observed that there is um, a segregation of the globin gene, the plant globins and the tissue specific globins. They have a different type of arrangement. Now, when we are taking an unrooted tree, you can see that these coming as a bifurcating node here. There is a bifurcating node. There is equal evolution here where the globin soybean and globin rice, they are having a common ancestor, which is not the case in this particular rooted tree. So this two have become one clade, and this has become one clade. And again you can see this neuroglobin and myoglobin, they are again separately placed. But what is the difference now between a rooted tree and an unrooted tree? In the rooted tree, you are able to understand the evolutionary relationship between the various mm, uh, samples which you are examining. But here there is no clarity whether each of them is segregated on their own, whether there is any common ancestor for these type of genes. Is there a common ancestor here? We have no idea. Whereas in the case of a rooted tree, you always have a common ancestor which is there. Okay, so what is the difference between a rooted tree and an unrooted tree? A rooted tree will try to identify the evolutionary aspect, evolutionary relationship, whereas an unrooted tree is just giving the relationship between the nodes, whether they have similarity or they are not having similarity. Clear?
Yes, ma'am.
Yeah. Now let us see how to calculate the number of rooted trees and unrooted trees. There is an equation by which you can calculate what are the number of possible trees, unrooted trees which can be formed from one particular operational taxonomical unit. And this, you can see, if there are, this is a mathematical formula. This is only for our understanding, to see that how many possible types of rooted trees, which is unimaginable for us to keep drawing these many number of trees. So these, depending on the number of samples which are there, or depending on the number of uh, parameters which you are observing, there can be n number of trees which can be constructed, both rooted trees as well as unrooted trees. So this, we can imagine writing one tree for a rooted tree or an unrooted tree, or three trees maximum. We can write about 15 rooted and three unrooted trees with little bit of effort. But it is humanly not possible to discuss or write the uh, so many number of trees if the number of operational units are increasing. So, and after drawing all these types with so many types of trees, what is the best tree you have to pick up? What are the chances that the tree which you have constructed, which of that tree is the optimal tree for explaining the similar relationship between the sequences of our interest? So in order to simplify this process, we have the methods of tree construction. So if you want to, this particular equation is telling you that so many types of trees are possible. You can construct these many number of trees, right? You can calculate, there are some 8 into 10 to the power of 12 trees. Can anyone write how many rooted trees are there or how many unrooted trees? It is not practically possible. So we take the help of some tools or softwares which divide, or which make it possible for us. We apply some mathematical through mathematical algorithms. Yesterday when you are discussing cluster alignment, madam was telling about dynamic programming, progressive alignment. So these are all the background work which is going on to give you the final output, give you a nice tree with all the values and with the numbers which are there. So to get to that, we have to use a different type of tools, just as to see the comparison of sequences or conserved sequences. You have taken a ClustalW tool, or other types of tools, mapped, or other types of tools were shown to you yesterday. Now, similarly, if you want to construct a tree, what is the background work which you have to do, or which the tools have to do? So there are many tools which are there for the tree construction. Okay, so all these trees which are there for the tree construction, they are broadly divided into uh, different types of methods. One is called as a distance matrix method, and as the name indicates, this is basically telling us about the distance. Distance means you have seen that there are some numbers which are written on the top of the branches. Those numbers are indicating the distance with which one species differs from another. These numbers are what I am talking about. What you are seeing here, these are the numbers which I am talking about. They will be telling us, if you see these are equal, having the same number, so you have same length, but this number is slightly more, so the branch length is more. This number is less, so the branch length is less, and so on. So this matrix, that is a distance matrix methods. These are using the calculation, taking the number of sequences into account, number of nucleotides into account between different sequences, and finding out how many of them are similar, and based on that can we come to some evolutionary relationship understanding between the sequences under study. So distance matrix method includes the first one, that is UPGMA. This is a very traditional and very time tested and a very old method of constructing the phylogenetic trees. What is UPGMA? UPGMA stands for Unpaired Group of Arithmetic Mean. Unpaired Group Method of Arithmetic Mean. U unpaired, UP is unpaired, G is group, M is method, A is arithmetic mean. All of you understand what is a mean. A mean is an average divided by the total number will give you the mean. If you have 10 residues or 40 residues, in that 40 residues, if you are taking that and you have 10 such cases, you are taking the total and dividing it by total number of samples, that becomes the mean. So what this UPGMA method is, the UPGMA method is taking the arithmetic mean. We will see what it is. For an example, I'll just show you that in this discussion. So UPGMA method is just telling us the simple way of calculating the arithmetic mean. The next distance matrix method, it is called as a transform distance method, then a neighbor relation method and neighbor joining method. The neighbor relation method and neighbor joining method, these are deciding upon distance between the nodes. Okay, if you are taking the nodes, which are the nodes in this particular thing, this is a node and this is a node, this is a node, like this. Now, what is the distance between this node and this node? Which are the two closely related nodes? For this, the immediate node is this one, whereas this is a distant node, because this node has one more node here, one node here, and another node here. So, if you see the relationship, this is a neighbor joining. Which are the nearest neighbors for the nodes? That is the neighbor joining method, which is the NJ method. Majority of the tools which are using these distance matrix based approaches, they will be using the NJ method for constructing a phylogenetic tree. Then coming to character based methods, this method is called as a parsimony method, where it is based on the characters which are being shared by different sequences. So how much important is that particular character which is under significant study, that will divide that as unweighted parsimony or weighted parsimony. Okay. Now let me see the tree construction using UPGMA. UPGMA is unpaired group method of arithmetic mean. Now what is this? Let us see here. These are five, here you can see that there are 5 sequences. These are the homologous DNA sequences. There are some 50 nucleotides here, and here you have a distance matrix which is constructed. How this distance matrix is constructed? This is constructed based on seeing the sequence of A, comparing the sequence of A with the sequence of B. So if you carefully observe these nucleotides, you will observe that there are some 9 differences between. The number indicates that there are 9 positions which are different. You can see that between A and B. If you see this CTC, this is the same. You have a difference here, A to T, G to C, there is a difference here. And if you see this particular next set of 10 nucleotides, these are all similar here. This next set of 10 are similar. This next set of 10 are similar. Where is the difference? The difference is here. What is the difference? You can see already one difference you have observed here, A to G, and the remaining differences are here, G to A, T to C. Then these differences are going to be, and here also you can see A to G, one difference is there. So like this, there are around, between A to B, the number of differences are 9. So A to C, compare A to C, A to D, and A to E. So if you compare this, you will be observing later when you uh, Let us, you can count the number and see if this number is really there, but right now for present, accept whatever I'm saying here. So pairwise distance matrix, there will be, I'm just telling you how you can spot the differences. So from A to B, there are 9 changes. A to C, there are 8. A to D, 12, and so on. If you compare again, B to C, there are 11. And for each of this, you are seeing and you are looking at these differences. If you observe this particular table here, you can see that what is the one which is having the least difference of bases. The least difference of bases is between the D and E. So this number is 5 here. Why it is 5? Maybe you can find a spot the 5 differences here. This one is 1, 2 is I can see here. Similarly, there may be some other position. There may be some changes which are there. So you can see one more change here, 3 and 4 here. So like this, there will be some changes which are there. So if you see that the D to E, there is only 5 basis difference. So this is the one which is the least one. So the number, we are grouping them as D and E as one set. Now, the next one, which is the one that is having the least difference, is the A to B. Okay, so A to, yeah, A to B is having the least difference. A to C, you can see A to C is 8. Here it is 5, here it is 8. A to B is 9, A to C is 8. So this is the one which is closest. Next closest is A to C. So A and C are grouped together. So A to C is having a bifurcating node, D to E is having a bifurcating node, and then we are observing the differences of A, C, D, E to get to this particular point. If you have to get to this point, then you need to see what is the relation between what is the next major difference after that A to B, this is 9, and the next one which is there is B to C. So like that, you need to go in that particular order and finally complete your phylogenetic tree. Observe that these groups which are given like this, they are indicated in the brackets. So those two sequences which are together, they will be indicated by bracket, and the next group which is sharing a common similarity is given another bracket, and all together they are covered in another set of brackets. So this itself is an indication which are the species. If you read this, try to read this kind of notation. If you come across in any phylogenetic tree this kind of notation, then you should understand that these are the, what does this notation signify? What which are closely related, they will be placed in immediate bracket. Those which have close relation, they will be placed outside this bracket with another closed bracket, and altogether they are enclosed in a bracket. So this is the comparison of the various distant matrix method. The UPGMA method is a statistically based method, and this can be used for morphological and molecular data, and it assumes that there is a constant rate of evolution. The transform distance method is another method which is based on an outgroup and the different rates of evolution with different lineages. The neighbor relation method, this is the pairs of species are separated by just one internal node. That's what I have told you. The tree is created with the smallest possible branch length. Okay, so now that we have seen how to make a tree, what are the types of trees which can be constructed, how to go about the analysis part, how to do the analysis. I have the sequences, I have some 5 6 sequences, and I don't know how to relate the construct a phylogenetic tree. What are the steps I should follow? So in that case, there are, these are the 5 steps which we can follow, or the process which you should follow, not the methods. The process which you have to follow is identify the sequence. The first step is called sequence acquisition, where you are going to acquire sequences. from where you will acquire sequences.
Nana,
Yesterday, yes, hello,
Nana,
hello, please mute
free
yourself.
వస్తుందని చెప్పు బ్యాచ్.
Hello, please mute yourself. Okay, so sequence acquisition means where you are acquiring the sequence from. What are the various sources? Yesterday, in the ClustalW program, when you are doing multiple sequence alignment, you have been shown how to get the sequences. You can copy paste the FASTA sequences, or you can download them into one common file, and then you can download them into one common folder, create a file, and then upload those sequences. That is called as sequence acquisition. Now, what kind of sequences should you acquire? Taking up sequences is one aspect. If you want to do phylogenetic analysis, what are the sources or what are the resources from where you can get those kind of sequences that can be subjected for analysis? So here you can take nucleotide sequences, or you can take RNA sequences, RFAM, or ribosomal database for ribosome sequences, or you can take the sequences from PFAM. PFAM is the protein family database. So you can do phylogenetic analysis of DNA sequences, you can do phylogenetic analysis of RNA sequences, you can do phylogenetic analysis of proteins. Okay, fine. Once you have acquired the sequence, then you know the tools you have to do the sequence alignment, because the tree construction depends upon the alignment. Without doing alignment, you cannot do the tree construction, because alignment will tell you what are the similarities, what are the differences, how many nucleotides are similar, how many amino acids are similar. That is given only when you align all the sequences. So you have to do multiple sequence alignment. After doing multiple sequence alignment, you need to do the modeling, which is the best suited tree construction model for that. Then you have to do a tree building using different models, that is tree for the phylogenetic tree models, that is PAUP, Philip, Mega, Tree Puzzle, Mr. Base. All these are different softwares or different tools which you are going to be working with, or which you can use to make your tree construction. After the tree construction is there, and remember here participants, I would like to tell you that each of these methods which are here, the tree building method, the POP, Philip, Mega Tree, what will be the background which they are going to use? The background they are going to use will be either the distance matrix method, or it will be the parsimony method. In majority of the cases, it will be the distance matrix method, which is most of these tools, they will be using the distance matrix method, and more precisely NJ method, that is the neighbor joining method. So once your tree is ready, then you have to evaluate, as I have shown you, that there are thousands and thousands of trees which can be generated. I have shown you the table. Based on the sequence number, you will be generating so many types of trees, and you have to see which is the best possible tree that can be used further, or which is the best possible construction that is possible. So you have to evaluate your tree. Evaluation of the tree is done by what is called as bootstrapping analysis. So what is this bootstrapping analysis? This bootstrapping analysis is going to compare the trees which are generated by all the different methods of tree construction. It will develop the bootstrapping technique will make a duplicate of your sequences. It will work on its own. You have given a set of sequences. It will take the, you have done a cluster alignment. What bootstrapping does is it is going to take a copy, make a copy of your sequences. It is going to devise, or it is going to run through the alignment process. It is going to apply the tree construction, and after that it is going to apply a statistical calculation to that, to identify which is the statistically best model which you can come out for a particular set of sequences. Clear? Yes?
Yes, ma'am.
Yes,
Did you
ma'am.
understand till now?
Yes,
So,
ma'am.
yeah. So now that
Yes,
we
ma'am.
have, yeah, now that we have uh, seen the methods, we have understood the methods which are important for tree construction. Now let us go further and try to work it out with some suitable examples. Shall we go ahead? Shall we go ahead by doing some analysis?
Yes, ma'am.
Yes, ma'am.
Yeah. All of you are ready. So I would like all of you to first log into your Galaxy, keep it ready and let me know. Or if you want to work it out later, I will just share slides to you. You can make a note of what are the steps which are involved, or I can show them parallelly what it is. So I have some issue here. Let me just resolve that. Then I can maybe show it to you in a different manner. Just give me 2 minutes. Okay. So whoever is working with your system, if you are working with Galaxy or if you are wanting to work with Galaxy, you may let me know. Otherwise, I will just go ahead. So what is it that we are going to do? As I have told you, uh, the analysis part, first we have to get our sequences out, and after getting the sequences out, we have to do a sequence alignment, multiple sequence alignment. After doing a multiple sequence alignment, we have to construct the tree. After constructing the tree, we have to analyze our tree, right? So first step for us is, let me share the screen. So first, is the screen sharing?
Yes, ma'am.
Yeah. So what should we do first? Let us go to NCBI. NCBI. Okay. Let us work with nucleotide sequences first. Okay. So let us go here. So I'm just giving some name. For instance, I will take some random protein. For instance, you can say amylase or any such example. Okay. Okay. Right. So here you can, as I discussed yesterday also, let us see whether we could get different species or different examples. So for the demonstration purpose, I will take just click on these sequences. Let me take different species. Maybe we can change it to some other example. You can try out here. This is human pancreatic amylase. I'm taking more number of sequences because if you want to do clustral alignment, you should be having more than 4 sequences or 3 sequences. So I am taking different types of sequences. Okay, I have selected this sequence. You are all able to see it, right?
Yes, ma'am.
Yeah. And then go to yesterday you have been shown how to send a file. So instead of downloading every sequence, since we are going to open this in Galaxy, we will just send it to the file, select FASTA, and then create the file. Now this file which has been clear, this would be in my downloads. Okay, this file will be in my downloads. Okay, next. Is the Galaxy screen sharing?
Yes,
Hello?
ma'am.
Galaxy screen is sharing?
Yes, ma'am. Yes, ma'am.
Yeah. So now I have already done an example. Just I will already I have logged in. So here it will show that I have logged in here. Okay, I have logged in here. So it would be showing me this. So I hope all of you have logged in. And then what is the first step? Let me go to my slides. My slide is sharing, right?
Yes, ma'am.
Yes, ma'am.
Yeah, so now let me show the steps what I'm supposed to doing here. How to go to the Galaxy. Yesterday also you have been shown, so that is the same repetition what we are going to do now. So here the step one, okay, step one is you have to log in, then you have to upload your sequences. The screen is visible, or should I put it in screen share mode, slide share mode?
It is visible, ma'am. Can
Okay.
continue.
So this is step 2 is here. So in the step 2, what are we supposed to do? We are supposed to choose our local file. Okay, so I'll go back to the Galaxy and I will go for upload. Okay, choose local file. I have downloaded this one now. I told you it will be there in my downloads. So this file is there in the downloads. Okay, select the file and click on open and click on start. Okay, then you close it. Is it come? Yeah, it has come here. Can you see this? If anyone is coming for the first time today and you haven't seen this tool yesterday, so initially when we upload the sequence, it would be gray and then it will turn orange and then it will turn green. So green indicates that it is the job is done. Okay, so all of you know how to do this, right? So now let us go back and see what is the next step. We have completed step 1 and step 2. Now let us go to step 3. What is it that we have to do in step 3? In the step 3, we need to uh, search the tools. Okay, we have to go to tools. Why we have to go to tools? Because we have to align the sequences. Our sequences are only in the FASTA format and we have not done any alignment. So we have to go back to Galaxy and get the tools. So once you go to the Galaxy, coming to step 4, what is it that you have to do? You have to select the MAFT. MAFT is the alignment software. Yesterday, you have been given the list of uh, multiple sequence alignment tools like Clustal, Omega Clustal, and along with that, MAFT also is a tool based on the uh, algorithms or based on calculation what it does to do the sequence alignment. So all of you should remember whether you are using a Clustal program or a ClustalW or a MAFT or T coffee, any of these methods which are there, they do some background calculations and then give us a beautiful alignment. What is happening behind it is not possible to discuss in this session, but what happens there we can discuss some other we can be seen in a later point of time. But you need to understand that the MAFT is one of the multiple sequence alignment tool. So you have to go to the toolbox. I have given here what you have to do. So first you have to select your tool parameters. In the tool parameters, you have to give all these inputs. You have to change some settings there, and then you have to click on the run tool. Okay. So let me go back to my let me go to the Galaxy. I'm going to this. Just I will increase this font. Okay. So tools here, go to tools and type as MAFT. Okay. So you can get multiple tools using the MAFT program. So don't bother about all these tools. We are not still reaching that level now. Let us first understand the basic thing. So first you choose the first one, that is the MAFT multiple alignment program. Click on that. It will take you to this particular window. Okay, it will take you to this particular window where you are having the parameters. So you can see input batch. What is the input data you are giving to this particular tool? The input data you are giving automatically it identifies the file which you have downloaded in the FASTA format. Okay, you can see what are the accepted formats here. FASTA is the only accepted format for this particular tool. So you have to give only that particular tool. And what is the other input parameter I have told here? The type of sequences. So type of sequences, which is here, it is auto detect. And here the second input input which we have to give here, it is the MAFT flavor. That is how it is going to do that. So by you can use this particular tool which is there, how the MAFT is going to do on that. So you have why I'm selecting that itself, because if you click on this arrow here, you will see that there are many of them. Okay, and you can see that one is very fast using a progressive method, and there are slow iterative refinement method. So these are different methods which the MAFT tool is going to use. There are multiple tools like this. You can select different tools. So today we are only using this particular tool. Okay, MAFT flavor. That change you have to do in this MAFT flavor. If here types of sequences, what is the sequence we have given? We have not given amino acids, we have given nucleic acids. Otherwise, if you forget also, no issue, it will do auto detect. It will select on this sequence even if you don't forget to change it to nucleic acids, it will auto detect. So keep auto detect on so that you don't miss out on anything. And then is there any other thing we have to do the correction? So output format we require. So this is the output format. This is the only option we have based on our selection. You have Clustal output, Philip output. So we are selecting FASTA output here. And then I'm going to click on the run tool. It will take some time for the run tool to be done. So meanwhile, let us see what is the next step. We don't need for it to complete that to go on to the next step. So once the alignment is done, later you can visualize the alignment, and then we can go for the tree construction. We have to use the tree construction using another program which is called as the fast tree. So this fast tree is, again, you can see that you go to the toolbox and type as fast tree, you will come across different tools which are there, and this toolbox requires some parameters which are there. So what are those parameters? Make a note. One is selecting the type of alignment, whether you are doing a protein alignment or a nucleotide alignment. And then there is a model there because we are doing a tree construction. There are some inbuilt parameters which we are going to do, which the tool is going to perform. So uh, we are not I mean concerned with that, but you need to do the selection of that. So that we will do, and you have to go to advanced options. In advanced options, you have to select constant rates, and then you have to turn off. Why? Because if you want to do the alignment by using a neighbor joining method, we are going to do the alignment using NJ method or the neighbor joining method. So we need to turn off this maximum likelihood option, which it takes by default. It takes this by default, so that we have to convert it to S. Okay, so let's remember this. What we have to do, change the alignment, type of alignment, model, then the advanced options, and then we have to run the tool. Okay, so let's go to our thing. Now this is done here. If you are interested, you can click on that. So you will be getting a icon which will tell you what is there, and you can save this, or you have uh, a visualization here. You can visualize your sequence as we have done yesterday. You can use, if you click on this visualize button, you will get the multiple sequence alignment, and you can this is not logged in. This is logged in. Okay. Anyway, so you have to um, anyway, so here this is the parameters which we have set. If you want at a later time point, and after the workshop is over or this course is over, later you want to work on your own, your history if it is there, or you want to work it afterwards, you want to see what are the options you have set up here, you can select on this button. So now that we are done with the map, let us go next. Uh, what is the tool we have to give here? The tool which we have to give here is the fast tree. So let us go to the fast tree tool. Okay, so fast tree tool is there. So this you can see, this is building maximum likelihood phylogenetic trees. So we don't want maximum likelihood tree, we want NJ. That's why we are switching off it when we go to that parameter. Okay, so I'm clicking on this fast tree. It will take us to this particular window, and automatically it will take up the input file, the FASTA file. What is it? It's supposed to work. It will uh, take up this particular data. Otherwise, if you want to change that, you can use this particular scroll down. You have all these lined up on this. So you can, if you are missing on anything or you want to change your parameters, you can select a data set. Right now I'm selecting this one. By default, it will take the parameter or the file format which it requires, so we don't need to do anything there. Since it is a nucleotide, I told you we have to do the alignment here. So here we have to take the nucleotide alignment. So select this one and select the nucleotide here. And here I told you we have to make some corrections. Nucleotide evolution model. You just select this Jukes Cantor model, and coming to advanced options, go to advanced options. Here you have advanced options which are coming. You have constant rates. Use constant rates here. That is what I have shown you in the PPT also. You have to use the constant rates there. And then here turn off maximum likelihood is there. Just use this button, it will become open, it will be clicked as S. Then don't change any other option and click on the run tool. Okay, so then let it go on. We'll go to the next step of our analysis. So we have completed this particular step, step 5, and then come to the step 6, which is our step, that is the tree visualization. How to visualize your tree, whether the tree has been constructed or not. So to see the tree construction, we have to go into the toolbox and click on the type as a new VIC. So you will be getting these options here. You are all following, or should I slow down, students?
Following, ma'am.
Shall I stop for a while or shall I continue? Okay, so now tree visualization is there. So let us see whether our activity is over. So our activity is done. So once this activity is done, this again is the same way you can do visualization, whether a tree is constructed or not, and you can save your tree in different formats, and also you can check here, run the job again by changing the parameters. You can see here, and this is the phylogenetic tree visualization. So you can, I don't know, this is not coming anyway. It's okay. If you are able to get it, it's fine. Some issue here, I think, with my Galaxy. Okay, so this is the visualization part. You can see that some tree has come up on this window. Uh, okay, so our tree is ready. You can construct you can see your tree. If anyone has completed, you can share the tree and share your screen. Show me what is the output you have got. And also here, any visualization option is there? Okay, there is no visualization option here. Okay, so that is what we have done so far. So we have reached step 6. So in our previous step here, if I go to this slide, we have done sequence acquisition, we have done sequence alignment using MAFT, and then we have done the tree building by using the MAFT program, and also seen the visualization of the um, tree. Anybody could get the tree? Any tree, doesn't matter. Whichever sequences you have taken. Yes,
Yes, ma'am.
I don't know. Is this sharing or not? I don't know.
In CBI site is open, ma'am. Your system you are able to see.
I don't know where I went. Now it's
The
come.
screen is visible, ma'am.
Now it's come. Galaxy has come.
Galaxy has come.
Yeah. Okay. Okay. Now, what is the next step we have to see? Step 6, we have completed tree visualization, and next we have to see the analysis part. So analysis part, that is called as the bootstrapping technique. As I explained to you already, what is the technique which is going to analyze all the trees by different methods. When you see the bootstrapping, you will realize that it does the tree construction by different methods and then gives an output. So let us go to this particular step here. So what is it that we are supposed to be doing for this? You have to select the IQ tree here, phylogenetic evolutionary tree construction for multiple sequences. So you have to select your input file. For this, it is the FASTA file. It will be giving you the options. Then you have to specify the DNA sequence, and then you have to go to the modeling parameters. In modeling parameters, you have to select the rate heterogeneity here, and then you have to select the bootstrap parameters, and then you have to analyze the sequence. Okay, so here I'm going to put it as IQ tree. So when I say IQ tree, there are many tools which are coming up, because it does more. You can see that there is a ultra fast bootstrap phylogenetic tree with IQ tree. So it is a tool which can align, or it will join different types of sequences. So I'm selecting IQ tree. I have selected the IQ tree here. So what is the next step we have to do? We have to change the tool parameters. In the two parameters, first thing I told you, specify input alignment file in the Philip format or FASTA format or Nexus, Clustal or MSF format. So since we have this particular data, you can put it the mapped on data 12. Okay, that is what I have shown you here in the previous slide, mapped which we have taken, and then you have to select the type of sequence, specify sequence type, or already it has by default it is taking as a DNA sequence, and then go to the modeling parameters. In modeling parameters, you select rate heterogeneity. In rate heterogeneity, here it is given select the options which are there here um, in rate heterogeneity. Okay, you have the in rate heterogeneity, you have to select this particular parameter, write maximum likelihood site rate. So this one you have to put it to yes, select, just click this button, and then go to bootstrap parameters, select ultra fast bootstrap, and here you have to give the number of replicates. Let us put it as 1000. Later you can change if you want to change the number of
Hello, Padma ma'am. Hello, ma'am. Padma, ma'am.
పద్మ మ్యామ్ వాయిస్ వినబడట్లేదు మేడం.
A small technical issue, ma'am will join with us shortly. So by the time, if anyone has started to do with Galaxy, maybe you can work on that one until whatever step you have done, you can just go through it. Just hold on for 1 or 2 minutes. She'll join us back. So, how many of you have registered Galaxy? You can just put Y, capital Y in the chat box. And hope you're working with that one. Same thing. Yes. Okay, so many have started to do the work with Galaxy. That's really good. Ma'am has given some 6 to 7 steps till now, right? So how many steps you have been able to do till now? Step 1, step 2, or 3, 1, 2, 3, you can just put. I can understand your till which step. Maybe many of the other things. Oh, Managnya Sandhu, very good. 4th step, 6th step, very nice. You're following it. Very good. Did anyone was able to see the output of your visualizing the tree? Just put Y if you have seen the tree. Yeah, Lakshmi Nikita says Y, Nandini Y. Okay, good, fine. So, okay, so what about the others? And one kind information I would like to share. We are trying to sort out the list of participants who have registered and also paid the fees. So by tomorrow morning, I'll be finalizing the list. And 1 or 2 small issues are there, which I'll approach you personally to get it sorted out. And those who have registered, paid, and attending the classes regularly, giving the attendance every day, will be eligible for getting the certificates. So registration is mandatory. So please make sure that only the registered participants and participants who have paid will be eligible for certificates. So I'm just informing you it now itself.
Hello?
Ma'am, I can hear you. Yes, ma'am. Please,
Yeah,
ma'am.
yeah.
Continue, ma'am.
Okay,
Yes, ma'am.
okay. So which step? Okay, I'll start sharing my screen. Something else is coming. Is the screen sharing?
Yes, ma'am.
Yes, ma'am.
So this is the last step we have to do. Just a second, some other issue has come. Stopping the sharing, it is some other window is coming. I will just now it's fine. Now it's sharing.
Yes,
This now,
ma'am.
this one.
IQ tree galaxy is visible to us.
But this thing is, I'm unable to take out this side. This particular one is coming. Okay. Okay, so okay. Sharing is gone now.
Yes, ma'am. It is visible, ma'am, galaxy.
Yeah, we'll have visible, but I'm getting this particular screen on the right side. I'm unable to access those links there. It's not visible to me. Okay. Okay, so where are we now? Yeah, PPT sharing. So we have click on the run tool, and then your job is done. So now here we have the maximum likelihood distance matrix. You can visualize your result there. You can see the data there. So all other this is all other information which the bootstrapping method has done. So if you want to see this data, you can look into this data. This is not our purview. Okay, so let us see the tree construction. Maximum likelihood tree. I will see. This is the distance matrix. So a distance matrix means it is only going to give the list of values here. So let us take only the maximum likelihood tree which is there, so that we can see the tree construction. Okay, so phylogenetic tree visualization is there here. I'll click. Oh, this is also giving the same issue, but if anyone else, I'll show the slide which I have shared here on a different note. So this is the for a different set of sequences I have done earlier to just for showing you demonstration what the tree construction is. So this has also been followed the same methods what we have used in the present demo class. So here you can see that there is a close relationship with whatever these sequences are. These two are grouped together. This is a totally out group. This is totally out group. All of these have no relation with each other. They are all distance. Remember that this is by neighbor joining method. So neighbor joining method is going to give us the relation between the nodes which are closely related to each other. So you would be getting this kind of output. Here maybe I should log out of this and then log in. I should be able to get out. Let me check.
Padma ma'am, once you can share the slide and tell to them the
No, that's
result,
what I
because
shared. I shared the slide is not visible. This
no,
slide.
no. We were able to see only galaxy. Slide we did not see for the
This
result
one.
interpretation.
This now result is visible. Slide is
No,
visible.
no, galaxy is visible.
PPT.
PPT we didn't see.
So many windows.
Galaxy is visible.
PPT.
Now it is visible.
Okay. So this is the slide. But here you can see that the branch lengths are not given here. So that could be the reason when we are doing in the galaxy, when we are let us go back to our earlier galaxy screen is visible.
Yes, ma'am.
Yeah. So what is the alignment we have done? We have fast tree set up. Let's see here whether we have given an option to show the branch length. Let me see whether it is. There is one option which will come, I think, in the new week. Did we do new week method, new week step? New week step, I think I missed it, I think. Okay. So this one, here, I'm going back to this new week step. I have not shown you, I think, in this. Did I show new week step? I think output
No,
is,
ma'am.
yeah, new week is here, but this is our different data set, so mostly, yeah. So in this new week format, actually I have shown you in the slide, but when I was doing here, I didn't show, I think. So this is basically you want to visualize the branches and all. So in this, I have also shown on the PPT this new week one. Is this PPT sharing?
Yes, ma'am.
Yeah. So here I have given the option that this have not written. Yeah, that's why it is missed. Okay, so new week is one option which we have to see to display the branch length. So for that you have to type in the toolbox, go to the new week and select new week display, and you have to select which file you want to do that. So here I have taken the mapped well. Okay, and then here you have branch support. Here you have to show display branch support, and here in branch length you have to select display branch length. Okay, and here choose an output format. You have to select the PNG format. This is what I have missed. I think that's why maybe I wasn't getting whatever it may be. Okay, so run the tool. So you will be getting your data on the new week format. Showing red. Yeah, so you can see this picture, students. Your participants, you are able to see the screen, new week display.
Yes, ma'am.
So I will see here. These are the branch lengths which are given. You have seen this is the different. These two is, you have seen some numbers written on this. This is 0.31 and this is 0.25, where the 0.31 is there, the length is little longer, and 0.25 length is little shorter. So these two sequences, they have some amount of similarity. So these are put as a bifurcating node, and this particular one is at a distance. For everything we have, there is a value which is given on the top of this branch. So this is called as the branch length, indicating what is the closeness of the sequences between them. So if you see these two sequences, this is having 0.16 and 26, and this one is totally out. It is having a distance of 0.43 here. Whichever is coming out of the group, you can see that this is 0.46. So here you are not you are getting a tree construction where the samples are all we have taken diverse samples. If you remember, whatever I have selected from the from the NCBI site, we have taken diverse species. If NCBI site is open, let me go back to the NCBI site if it is open. I think it was in a different window anyway. So what I want to tell you is, yesterday also I was telling you, whenever you want to appreciate the evolutionary significance, you need to take different types of species, not the same species, or as we have seen, the same gene from different species, or same gene coming from a human, coming from a fly, coming from another species. So you would be getting a very clear picture for learner purpose, for depicting purpose, or for understanding the significance of the phylogenetic tree construction. So this is your final output, which you can get by using the Galaxy tool. So here Nubic display means you need to select your tree alignment. Earlier I have selected the MAFT alignment, so it did not give me this. So you have to select the fast tree output for the as an input file for the Nubic, and select the branch support and also the branch length. If you don't select this and select the type of output which you want, whether you want a PNG format or a SVG format or a text format, then you will not may not be able to visualize the picture, or you may not be able to download and save it for your work, or you want to put it in some project report or whatever, you may not be able to save it. So you better use the Nubic format and save your output in the PNG format, so that you can print it out and use it for your further work. Okay.
Padma ma'am, there is one participant, Srinidhi by name. The query is there in the chat box. Please, maybe she's got struck somewhere.
For the IQ tree tool, we could hear you saying till specifying number of bootstrap left gate, after which we are supposed to change no other parameter, just to change the replicates. Yeah, that is Arena. That is because in the Nubic format, you select the input file correctly. You select your fast tree output. I told you just now that if you select the MAFT output, you may not get a Nubic output. You select the tree construction. The tree construction is done by using the fast tree format, fast tree tool. Use that output from fast tree tool and then proceed further. You will be getting the image. Srinidhi, did I clarify your doubt? Okay.
Ma'am?
Arena, were you able to work?
Ma'am?
Yeah, Mahesh, tell me, Mahesh.
Ma'am, can you show me the sixth step in PPT?
Which one? Sixth step.
Sixth step
Which
in
one?
PPT.
Fifth.
Sixth one.
Sixth one.
Six.
This one, tree visualize. You are able to see?
Yes, ma'am.
Yeah, tree visualization. Here parameters, just now I showed you this, right? Branch support, display branch support, display branch length, and then click on the run tool.
Okay, ma'am.
So that is how I have shown a very brief overview of the phylogenetic analysis and how I have just shown you with nucleotide sequences. Maybe you can, though it is outlined to show protein sequences and nucleotide sequences, due to constraint of time, I could not show the protein sequences. Maybe you can try it out, and whenever I come back next to the class, if you have any queries regarding that day we will be able to solve it further. Okay, so that's all from my side. If anyone has any uh, queries or anything clarifications, I will be happy to complete that so that we can wind off today's session. Anybody is facing problem by using the Galaxy tool? Please respond yes or no. Hello? Hello?
It's clear, ma'am.
Okay. Are you all comfortable with the Galaxy?
Yes, ma'am.
Okay, then Vanita, can you post the attendance so that we can wind up?
Yes, ma'am.
Yeah, protein also you follow the same steps, but go to NCBI site, and in the NCBI site you select the proteins instead of the nucleotide, and follow the same steps of downloading the file and uploading the file. It is the same, only your source information will be different. So the attendance sheet has been posted in the group. Student participants, please complete the attendance sheet. Access is not provided, it seems. Anita?
I have checked, ma'am. I have given access.
But they are saying access is
Just
not
you
provided.
check it one more time. Now I've given, I've saved it. One minute, I'll check it out. Just try this link one more time. No.
Here it is asking. I tried opening. It is giving email
It's
or
not
something.
giving access?
No email. I have to give email and check.
Yesterday it was working. I changed it today and gave access. But
Even I am
still...
you need access. It is saying.
And Padma ma'am, ఇది ఒకసారి ట్రై చేయండి. I opened the link again second day only. Let me see whether it's working out. మీరు ఒకసారి ట్రై చేయండి పద్మ మ్యామ్.
ఇదా ఇప్పుడు పెట్టిందా?
ఇప్పుడు పెట్టిన లింక్.
అది వస్తుంది. అటెండెన్స్ డే 2 వస్తుంది.
Maybe people do give the attendance date wise and sort it out.
21/2 ఉంది. అది 21/1 ఉంది డేట్.
మారుస్తాను అవి మార్చి.
Excuse me, ma'am.
యా మనోజ్ఞ.
మ్యామ్ అది నిన్నటిది లింక్ ఓపెన్ చేస్తే యూ ఆల్రెడీ రెస్పాండెడ్ అని వస్తుంది మ్యామ్.
Okay. Now you recheck again. Sorry for the inconvenience. Recheck again.
Ma'am, it's showing attendance of day 2. This is day 3, right?
I'll change. It gets registered with today's name, right? Still same, Refresh your page and try.
Or for today, let us take from the teams that attendance list for today. And tomorrow we can give a new form to those who are there today.
Ma'am, is it possible to conduct the sessions on Zoom or Google Meet?
No, because Google Meet we can't do the recording, one thing. And Zoom, it will not, I mean, we don't have, I think we have Zoom account, Microsoft Teams account for our college. So Zoom will not support more than 45 minutes. We don't have the Zoom which supports longer duration.
Okay, ma'am.
Attendance is working out or not?
No, ma'am.
No, 53 responses are there. Not working. But how it became then?
Yes, ma'am. Now I got it.
It is working. 54. Your responses are increasing. Refresh your page and try. Or close the link again, you open it.
It did, ma'am. Thank you.
Yeah, so on this platform, I would take this privilege to thank Dr. S. Padma ma'am for giving an elaborate and theoretical explanation as well as showing the practical way how to execute the protocol in order to retrieve the sequences and use Galaxy tool to upload all the sequences, and step by step very clearly ma'am has given the details how to construct the phylogenetic tree for the nucleotide sequence. So maybe you can start working on with a protein sequence which you can download it. So that would be the outcome of today's class. So thank you so much, Padma ma'am, for such a nice, wonderful session for today. Thank you so much, ma'am.
Thank you, Anita.
Thank you, ma'am. Thank you, Padma ma'am.
Okay, Kamala, thank you. Thank you all for your patient hearing for 2 hours. Thank you all.